Photosynthesis. Photosynthesis nourishes almost the entire living world indirectly or directly.
Wow.
2 stages. Light reactions, which convert solar energy to chemical energy, and the Calvin cycle, which is anabolic. Light reactions overview is pretty much split water molecules provide a source of electrons and protons, of which dioxide is released as a byproduct. These electrons and protons are transferred using the light absorbed by chlorophyll to an acceptor. The light is able to be absorbed because of a light harvesting complex, which contain protein and pigment molecules bound together that act as an antenna for the reaction center complex. The reaction center also contains a primary electron acceptor, which is capable of accepting electrons and reducing the pair of chlorophyll A molecules, making it possible for electrons to transfer between energy levels. Chlorophyll A participates directly in these reactions, and it is able to receive the energy because of chlorophyll B. This shows that violet, blue, and red light work best for photosynthesis since that's the light that chlorophyll is capable of capturing. This is confirmed by the action spectrum, which profiles the relative effectiveness of different wavelengths of radiation in driving the process of photosynthesis. However, carotenoids broaden the spectrum of colors that can power photosynthesis, but they also have the more important role of photoprotection. They do photoprotection by dissipating extra light so that there's no damage to chlorophyll or so that there's no formation of oxidizing molecules. When a pigment molecule absorbs a photon, it's transferred between other pigment molecules, kind of like a human wave at a football game. Afterwards, they even glow, which is called fluorescence. So the electrons that are transferred to NAD+, which is nicotine adenine dinucleotide phosphate. This is reduced to NADPH by adding a pair of electrons and a hydrogen ion. The NAD+ temporarily stores the electrons and photons. Photosynthesis majorly takes place in the leaves of plants, which contain mesophyll tissue, whose cells contain chloroplasts. Chloroplasts contain thylakoids. Thylakoids transform light energy into ATP and NADH. Thylakoid membrane has 2 photosystems that cooperate for the light reactions, and the steps are split between photosystem 1 and photosystem 2. Photosystem 2 is named second for order of discovery but forms first in the light reactions. Light energizes these 2 systems to synthesize ATP and NADPH. The flow of electrons through these systems is key for the reactions. This is called linear electron flow, and there are 8 steps involved. An electron is boosted to a higher energy level in a pigment, making it excited. This occurs until it reaches the P680 pair of chlorophyll A molecules. 2. The excited electron is transferred to the primary electron acceptor. The P6080 becomes the P680+. Next, 3. An enzyme catalyzes the splitting of a water molecule into 2 electrons, 2 hydrogen ions, and an oxygen atom. Then, the electrons are given to the P6080+ pair. The hydrogen are released into the thylakoid lumen, and the oxygen quickly becomes a diatomic, or O2. For step 4, each excited photoelectron moves from PS2 to PS1 via the electron transport chain. The electron transport chain is made of the carrier protein plastoquinone and the protein plastocyanin. 5. The electrons release heat as they fall to a lower energy level, providing energy for the synthesis of ATP. Heat and energy. 6. Meanwhile, light energy has triggered the excitement of the P700 chlorophyll A electrons. This creates a hole that, when filled, will become P700+. Photoexcited electrons have been passed through a series of redox reactions on a second electron transport chain through a protein ferredoxin. 8. The enzyme NAD+ reductase catalyzes the transfer of electrons from FD to NAD+. 2 electrons then allow for its reduction to NADPH. Next is the Calvin cycle, which has phase 1, carbon fixation. It incorporates each carbon dioxide molecule one at a time by attaching it to a 5-carbon sugar called ribulose biphosphate. The enzyme that catalyzes this first step is RUBP carboxylase, or Rubisco. Results in a 6-carbon intermediate that is so unstable it immediately splits in half, forming 2 3-phosphoglycerate molecules. Then, phase 2 is reduction. Each 3-phosphoglycerate receives an additional phosphate group from ATP, becoming 1,3-biphosphoglycerate. Then, represented by NADPH, reduces the molecule, making it lose a phosphate group, thus becoming G3P, which is a sugar. These molecules are formed for every 3 carbon dioxide molecules. One of these 6 will be used by the plant, while the other 5 will be recycled to regenerate RUBP. Phase 3 is the regeneration of the carbon dioxide acceptor, which is RUBP. The carbon molecule of the 5 molecules are rearranged into 3 molecules of RUBP. Cellular respiration. Cellular respiration occurs in 3 stages, but biochemists normally recognize stages 2 and 3. The first stage is glycolysis, which is anaerobic. Glycolysis means sugar splitting. Step 2 is pyruvate oxidation in the citric acid cycle. Step 3 is oxidative phosphorylation and electron transport and chemoosmosis. 1. Glycolysis. Glycolysis, it means the glucose is split into 2 3-carbon sugars which are then oxidized. The remaining atoms are rearranged to form 2 molecules of pyruvate. More specifically, there are 2 phases to glycolysis: energy investment and energy payoff. Energy investment, the cell spins the ATP. Energy payoff, ATP is produced by substrate level phosphorylation and NAD+ is reduced to NADH. The net energy yield per glucose molecule is 2 ATP plus 2 NADH. No carbon is released as carbon dioxide during glycolysis. Pyruvate oxidation in the citric acid cycle. Pyruvate oxidation, pyruvate enters the mitochondrion via active transport where it is converted to acetyl coenzyme A or acetyl coenzyme CoA. Carried out by the catalyzation of 3 reactions. 1. Pyruvate's carboxyl group is removed and given off as carbon dioxide. 2. The remaining 2 carbon fragment is oxidized, forming acetate, and extracted electrons are transferred from NAD+, storing energy as NADH. 3. Coenzyme A is attached via its sulfur to the acetate, forming acetyl CoA, which has high potential energy, and this acetyl group will now feed into the citric acid cycle. The citric acid cycle is also known as Krebs cycle. First, acetyl-CoA adds its acetyl group to oxoacetate, making citrate. 2. Citrate is then converted to isocitrate by removing a water molecule and then adding a water molecule. 3. Isocitrate is oxidized, reducing NAD+ to NADH. Then the compound loses the carbon dioxide molecule. 4. Another carbon dioxide is lost and the resulting compound is oxidized, yet again reducing NAD+ to NADH. The remaining molecule is then attached to coenzyme A with an unstable bond. 5. CoA is displaced by a phosphate group, which is then transferred to GDP, making GTP. GTP is similar to ATP. It can also be used to generate ATP. 6. 2 hydrogens are transferred to FAD, forming FADH2 and oxidizing succinate. 7. Water is added, rearranging the bonds in the substrate. 8. The substrate is oxidized, reducing NAD+ to NADH and regenerating oxoacetate. 3. Oxidative phosphorylation, electron transport, and chemoosmosis. Electron transport looks like this. Electrons removed from glucose by NAD+ during glycolysis in the citric acid cycle are transferred from NADH to the first molecule of the electron transport chain. This is a flavoprotein. It is then transferred to an iron sulfide protein, then to an ubiquinone. It is the only member of the electron transport chain that is not a protein. The remaining carriers are protein called cytochromes. Each has a heme group. The last is site A3, and it passes its electrons to oxygen. Each oxygen also picks up a pair of hydrogen ions, which forms water. FADH2 can also be used to get electrons, but it yields 1/3 of the energy of NADH. Chemoosmosis. Via ATP synthase, protons flow back down their gradient. The ATP synthase harnesses the proton motive force to phosphorylate ADP. This forms ATP, an ATP energy drink. Skip oxidative phosphorylation and get what you really need.